Today, we're going to talk about enzyme. So an enzyme, it's a protein, but it's the class of protein that act as catalysts. So our proteins, remember, they did lots of things. They could transport molecules, they could be receptors, they could bridge between cells to hold them together. One of the classes was catalysts, and these are extremely important in chemistry. So when we think about making a rate go faster, making a reaction speed up, you need to either heat things up, increase the concentration, or have a catalyst. Well, say you're trying to digest food, you can have more food around, so it'll break down faster, but there's only so much space in your stomach. And you can't really heat your body up. You're stuck at 37 Celsius. Get too much hotter and, well, you don't make the reaction go faster, you just die. So we need a way to speed up reactions like breaking down food without heating them up. And so a catalyst is the next best route. And honestly, well, it's the best route. The catalyst makes the reaction go faster at the standard body conditions. How does an enzyme work? What's going on? It just lowers the activation energy. So I don't know, it's been a little while since 121, but you have some starting material, you have some product, and normally there's a bit of a hurdle to get over before you can do a reaction. And that amount of energy, that increase that you have to put into the molecule to get it to react, that's activation energy. You can cheat though. If you use a catalyst, that is, you just take a different route. You don't have to put as much energy in. There's an easier way. An idea for this would be try to jump onto the second story of a building. The chances are you can't just jump all the way from the ground to the top. But if there's a bunch of stairs nearby, you could go up them one at a time, make the same journey, it's just much easier. This is what enzymes help us facilitate. They do the reaction, but they do it in a different way so that each individual step is easier than the original method of doing it. Well, rates are what they are increasing. This happens all the time. Like we throw acid into reactions, make the reaction go faster. That was one of the most common catalysts in our reactivity section. Difference with enzymes is they are very specific. What that means is that they tend to react with just one thing normally. Occasionally they can be a little less specific, but when we throw acid in, all of our reactions had acid pretty much. Like it just reacted with everything. But if you have an enzyme that's designed to break up amylose, so amylase is the enzyme that we use for that, it only reacts with that. You can't put in sucrose, you can't put in cellulose, you can't put in anything else. It reacts with that one thing. This is amazing because most of the catalysts that we ever use in industry, if you have another chemical in there, they'll react with it and they usually destroy each other. You have to be really careful to keep them safe and contained and clean. Enzymes, they'll work inside your cell with literally every other biomolecule kicking around, and they only do the one job they're supposed to do. So they are remarkably specific in their tasks. There's a few exceptions to this. Things like P450s. These are a class of enzymes that exist in your gut. Their job is to react with everything. So when you eat food, you eat like a plant or something, there's a bunch of random aromatic molecules from the plant that our body doesn't have any use for. We've never evolved a way to do anything with them. We need to get rid of them, but they don't dissolve well in the blood, so they're just kind of stuck there. What a P450 will do is if a large, ring system, very nonpolar, comes in, the P450 will slap OHs on it, and it will just keep slapping OHs on these molecules over and over and over until finally it becomes polar. And now it will dissolve, go through bloodstream, our kidneys can urinate it out, and it goes away. So it's a very interesting exception. P450s kind of do the opposite. They work with most everything to just throw alcohol groups on molecules. Why this is important when it comes up, this is how most drugs actually get destroyed. A lot of the drugs we take, we don't want them to break down very quick. We want them to hang out for a while. Your body will slowly destroy them, and the P450s is usually what's doing that. So there's been a lot of study on P450s so that we can figure out how fast drugs will interact with it, make them last a little longer, or in some cases, make them last a little less time. You don't want a drug to persist for too long doing its effect if that means you can't control being able to stop it if it needs to stop. Well, there's a few things that alter enzymes. So we're going to talk a few things about regulation. You want your enzyme speeding up a reaction when you need that reaction to happen. But if you don't need the reaction anymore, you don't want your enzyme doing that. So there's a few things that can regulate it. Basically control the rate, or as it's also known, the activity. Basically control how fast it's doing its job. One of the things is just substrate. Substrate is the chemical that's being reacted. If you just don't have a lot of it around, then you don't react a lot of it. If you need more reaction to happen, add a bunch more substrate, it tends to go faster. So you can adjust your rate a little bit by changing the thing it's working on. There's also inhibition. Inhibition lets you actually slow the enzyme. Basically, depending on what enzyme, you can turn up and turn down, like a volume knob, how fast that enzyme works. And we'll talk a little bit about how you can alter it, but it allows you to slow down an enzyme when you want it slower, or turn it back up when you want it faster. There is inactivation. Permanently turn off. You can just destroy the enzyme. Your body makes more eventually, but sometimes we want the enzyme to just stop. This actually happens in your stomach. Peptidase is a molecule that breaks down proteins. We have it in our stomach to help break down the food we eat, get those proteins. Because our stomach has a thick mucus lining, which is sort of a long fat with sugars attached. It's actually what mucus is, if you didn't know. It's a fatty molecule with sugars to keep water stuck to it. But that protects our cells so that the peptidase doesn't get in and wreck up our stomach. But when it leaves our stomach and goes to our intestines, it could actually start digesting our intestines. And so the body actually releases another molecule right as it leaves the stomach that permanently destroys the peptidase. It breaks it so that it can no longer break down proteins. And so that is a form of inactivation. It permanently disables that protein, which means our stomach has to make new peptidase every time it wants to digest stuff. But it's energetically worth it. It's a catalyst, so it doesn't take a lot of energy to make it. It breaks down a lot of molecules that we can then use. And finally, one of the things that can regulate is product concentration. We'll discuss that with inhibition, but a lot of times the product, if you have a lot of it, can slow down the enzyme, either because of equilibrium favoring going backwards, or because it slows the enzyme by interacting with it.